Welcome back to Share Science, the weekly science brief for June 22nd, 2026. I'm Curie.
And I'm Derek.
Okay, so here's something wild. Mammals, including humans, might actually have the ability to regenerate lost body parts, but it's been switched off by evolution. And now, scientists think they found a way to turn it back on.
That's a pretty bold claim. Are we talking about salamander level regeneration here, or is this more of a first step kind of discovery?
It's definitely early days, but the findings are incredible. Researchers at Texas A&M developed a two-step treatment that redirects the body's natural healing process. Instead of forming scar tissue after an injury, this treatment encourages the body to regrow complex tissues like bones, joints, and ligaments. They tested it in animal models, and it worked.
So, just to clarify, this isn't about humans regrowing limbs tomorrow, right? This is still in the experimental phase?
Exactly. This is all happening in animal models for now, but the mechanism they've uncovered is what's so fascinating. They used two signaling proteins, BMP2 and FGF2, applied in a specific sequence to the injury site. These proteins essentially reprogram the cells at the site, flipping a biological switch from scar mode to regrow mode.
So, the ability to regenerate has always been there, hidden in our biology, but evolution decided to prioritize quick healing and scarring instead?
That's exactly it. Scarring is an evolutionary advantage because it closes wounds quickly, preventing infection and blood loss. But that quick fix approach comes at the cost of regeneration. What this research suggests is that the instructions for regeneration are still encoded in our cells, just suppressed. If this can eventually be translated to humans, the implications for medicine are staggering.
It's like rediscovering a superpower we didn't know we had. The potential for amputees, for people with severe injuries, or even for reconstructive medicine is just mind blowing.
Totally. It's one of those discoveries that makes you rethink biology entirely. If you're curious to learn more, search for mammal regeneration, BMP2, FGF2, Texas A and M 2026 to dive into the study.
All right, shifting gears, let's talk about our second story, the fentanyl vaccine. This one addresses one of the most devastating public health crises of our time, the opioid epidemic. Researchers at Scripps have developed an experimental vaccine that could prevent fentanyl overdoses by stopping the drug before it ever reaches the brain.
This is such a fascinating approach. The idea of a vaccine against a drug is pretty novel, right? Most vaccines train the immune system to fight off pathogens like viruses or bacteria, but this one trains the immune system to recognize fentanyl molecules.
So, how does that work? If someone who's vaccinated is exposed to fentanyl, what happens?
When the immune system encounters fentanyl molecules, it produces antibodies that bind to them. This traps the drug in the bloodstream, preventing it from crossing the blood-brain barrier. And if fentanyl can't reach the brain, it can't cause the respiratory depression that leads to fatal overdoses.
That's clever, but fentanyl is notorious for its chemical variants, the analogs and designer derivatives that keep popping up. Does this vaccine work against those too?
That's one of the most impressive aspects of this research. The vaccine was designed to recognize a broad range of fentanyl-related compounds, not just a single molecule. That means it could potentially offer protection even against modified versions of the drug.
The implications are huge, especially for people in recovery from opioid addiction. Relapse is such a high risk period, and having a biological safety net like this could save so many lives.
Exactly. It's important to note that this is still experimental and will need human trials. And of course, it's not a standalone solution to the addiction crisis. But as part of a broader harm reduction strategy, it's incredibly promising. You can search Fentanyl Vaccine Scripps Research Overdose Prevention 2026 to learn more.
On to our third story, which is one of those how did no one see this before discoveries. Researchers have found a surprising connection between blood cancer mutations and Alzheimer's disease.
This one really caught me off guard. It turns out that certain mutations linked to blood cancers can create overly inflammatory immune cells in the brain, which may help trigger Alzheimer's disease.
How does that connection work? I mean, blood cancer and Alzheimer's seem like completely unrelated conditions.
It starts with something called clonal hematopoiesis. As we age, the stem cells in our bone marrow accumulate mutations. Sometimes, one mutated stem cell outcompetes its neighbors and produces a large population of blood cells with the same mutation. This is common in older adults and has already been linked to cardiovascular disease.
But how do these mutated blood cells affect the brain?
Some of these cells develop into immune cells that can enter the brain. When they carry certain mutations, they become abnormally inflammatory. And chronic inflammation in the brain is a major driver of Alzheimer's disease, accelerating the buildup of amyloid plaques and tau tangles.
So these mutated immune cells essentially act as a kind of Trojan horse, bringing inflammation into the brain?
Exactly. This discovery has huge implications. For one, clonal hematopoiesis could serve as an early risk marker for Alzheimer's, detectable through a simple blood test. And it also opens the door to new treatments targeting these specific immune cells to slow the disease's progression.
It's a fascinating reframe. Thinking of Alzheimer's at least partly as a disease that originates outside the brain. If you want to dig into the details, search clonal hematopoiesis blood cancer mutations, Alzheimer's inflammation 2026.
All right, let's go from the microscopic to the cosmic. Our fourth story takes us deep underground to the Juno Observatory in China, which has just delivered its first major breakthrough in neutrino research.
Neutrinos are such a mind bending topic. They're these almost massless particles that barely interact with anything, but they're everywhere, streaming through us by the trillions every second. Detecting them is like trying to catch a ghost.
And Juno is one of the most ambitious efforts to do just that. It's a massive sphere filled with 20,000 tons of liquid scintillator, surrounded by tens of thousands of light detectors, all buried 700 meters underground.
And their first big result?
They made an extremely precise measurement of neutrino oscillation, the way neutrinos change between three different types as they travel. This is a critical step toward understanding the neutrino mass ordering, which has profound implications for fundamental physics and even the mystery of why the universe is made of matter instead of antimatter.
It's incredible how these tiny particles could hold the key to some of the biggest questions in science. Search Juno Neutrino Observatory First Results Oscillation 2026 for more.
Finally, let's talk about ancient DNA and how it's still shaping human immunity today. Researchers found that Denisovan DNA, inherited from interbreeding with this extinct human cousin, is actively influencing the immune systems of people in the Pacific.
The Denisovans are such a fascinating mystery. We know so little about them, but their genetic legacy is still with us.
Especially in populations in Oceania, where people carry some of the highest proportions of Denisovan DNA. This week's research shows that some of those genetic variants are directly involved in immune function and are still helping these populations adapt to their environments.
It's a beautiful example of how interbreeding with archaic humans wasn't just a historical footnote. It actively shaped our ability to survive. Search Denisovan DNA Immunity Pacific Populations 2026 to learn more.
And that's our week in science. From regeneration to ancient DNA, the world is endlessly fascinating.
Until next time, stay curious.